Then let's talk about the general concept or the general, way to do protein ligand docking calculations. So first time, we need to think about scoring functions. So the scoring functions are, in other words, called energy functions. And the energy functions are used to predict binding free energy binding affinity between protein ligand complexes. So for the protein ligand binding free energies, in general, non-bonded interactions are playing an important role. So the non-bonded interactions are important. So what are the examples of non-bonded interactions? So the non-bonded interactions are generally, we can say that, the three examples are the electrostatic interactions and van der Waals interactions and solvation-free energy, solvation energy. So these are classified as a non-bonded interactions. So in other words, so the bonded interaction means that, so the opposite must be a bonded interaction, right? And the bonded interaction means that, covalently bonded. So the, bond length and bond angle energies because of bond angle or dihedral angle, something like that. And so when you design when you think about the scoring functions, actually the scoring functions can be classified into two types. So the first one is an empirical scoring functions, and the second one can be called physics-based scoring functions. So the empirical means that they are based on experience or data, based on the data. But on the other hand, the physics-based functions are generally based on, physics laws, like Newton's law or something like that. And every program has different scoring functions. So as I mentioned, so the scoring function is an approximation of energy, right? Approximation of Gibbs free energy. So anybody can, develop their own approximations. So there is no, certain, gold standard. So there are many different approximations, and so there are many different, scoring functions. So let's take a look at the, empirical scoring functions. So the example of empirical scoring functions. So the empirical scoring function means that they fit parameters. They fit parameters to experimental data. So experimental data means that, experimental, binding affinity data. And we assume a functional form. We assume certain functional form based on intuition, and based on that, so we fit parameters to the binding affinity data. So one of the example of a empirical scoring function from Autodock is that, so the Autodock is one of the protein ligand docking programs. So one of the most popular protein ligand docking program, and actually you can download the program from this website, but it only runs on Linux, maybe. I don't know. So maybe it can run on Windows. So maybe I should check. So, but generally, so it runs on Linux, and they assume that the binding free energy is a combination of 5 terms. So the first term is that the delta G rotation multiplied by number of rotatable bonds. So here the N rot is the number of rotatable bonds. And so this term represents the loss of entropy due to binding. So if you imagine that, so when ligand is freely solvated, when ligand is just here, when ligand is just solvated in water, then it can freely rotate, right? But once ligand is bound with some, protein, but once the protein ligand is bound with a protein, but it cannot rotate freely. So which means that it loses its freedom to rotate. So which means that, I mean, it loses its entropy. So this term represents loss of entropy. And the second term is about the hydrogen bond. And the third term is, I mean, ionic interaction. So this term takes into account the hydrogen bond, I mean, based on the experimental data, and also this term generally approximates ionic interactions between atoms. So these two interactions are assumed can be thought as a electrostatic interaction. And also, I mean, they considered aromatic interaction. So the aromatic interaction is sometimes also called as a pi stacking or the stacking interaction. So the stacking interaction is, I mean, I'm going to talk a little more detail in the later, but it's a stacking interaction is between the special interaction between rings. So I mean, so aromatic rings are known to interact quite strongly to each other. So they introduced this term to take into account that effect. And the final term is about the hydrophobic interaction. So the hydrophobic interaction is also, this term is considering solvation-free energy. But in other words, I mean, but on the other hand, I mean, so the physics-based energy functions generally based on the physics, and they have a functional form based on the physical law. So for example, so here, I mean, if some program is designed based on a physics-based energy function, then the non-bonding interaction can be written like that. So here the I means that the index of atoms of ligand, and the J is the index of indices of atoms of protein. Right. So here the Lij is the distance between atoms in ligands and atoms in protein. And as you know, I mean, so as you can see, I mean, so this term has the shape of van der Waals interactions, I mean, as you have learned from the textbook. And this term is about the electrostatic interaction, right? Coulomb interaction, right? I mean, Q1, Qj, Qi, Qj, and Lij is the distance between atom I and atom J. So the physics-based interaction energy function is generally designed based on the physical law. So the parameters are generally obtained from quantum mechanical calculations, and they are not from experimental data. So the physics-based energy functions are clear and they have solid theoretical foundation, but they are not necessarily more accurate than experimental or empirical scoring functions, because as I mentioned, I mean, so the quantum mechanical calculations of proteins takes long time. So generally they are based on some approximations, or we have to use very crude quantum mechanical calculations and so on. So in short, I mean, so the better more physical description doesn't always guarantee more accurate results. So let's talk about the solvation-free energy. So what is solvation-free energy? So if we think about the biological system, usually a solute should be solvated into solvent, right? Solute should be solvated into solvent, and in biological systems, solvent means generally water, right? And proteins, I mean, our body, I mean, so most organisms are based on water, right? So our cell is filled with water, and all the proteins and ligands are actually solvated in water. So, but I mean, if we think about the physics, then the solute, so the solute itself, if we think about a solute is in vacuum, so if it is in vacuum, then it should be solvated, right? So, I mean, this energy is a solvation-free energy. And the solvation-free energy is, so in other words, you can think it as the energy required to transfer a molecule from vacuum to solvent, right? So the solvation-free energy is also should be considered during the protein-ligand complex because, so when ligand is bound, when ligand is solvated here, but when it is bound with a protein, ligand is bound with a protein, then it loses some interaction with water. I mean, so when it is solvated in water, actually, so it is interacting with water molecules, right? But let's imagine that so this one is bound with a protein. This one is bound with a protein. Then it should lose its interaction with water, but instead, it should form interaction with protein. Okay, so if new interaction with protein is stronger than the lost interaction with water, then this molecule will prefer to bind with a protein. But otherwise, so if interaction with water is stronger than interaction with protein, then the water and this molecule, then this ligand must prefer to stay, solvated. It doesn't like to be bound with a protein, right? So this is why we should consider solvation-free energy. And this solvation-free energy can be decomposed into three terms, and the solvation-free energy generally consists of three contributions, and the one is the electrostatic interaction, and the second one is a van der Waals interaction, and the third one is about the cavity formation. So the electrostatic interaction is, as I mentioned, so the electrostatic interaction between water molecules and solute molecule. So if you think about the this picture on the right side, and here the sodium ion is solvated in water, and this plus charge and negative charge are interacting with each other, attracting to each other through electrostatic interaction, right? And second, we can think about van der Waals interaction, van der Waals energy, and also there is additional energy cost for cavity formation. So the cavity formation means that so in order for this solute to be solvated, then a cavity should be formed in water, right? So there is an energy cost for this. So these three effects contribute to solvation-free energy. And let's briefly talk about PiPi stacking. So the PiPi stacking interaction is also very common in chemistry. So the PiPi stacking interaction, or in other words, it is called as a stacking interaction. And the stacking interaction is between two aromatic rings. So if two aromatic rings are oriented like this, like sandwiches or like T-shaped, here the one is on the planar and the one is standing in a perpendicular way, or one ring is parallelly displaced from another ring, then so it is also can be considered as so they too may have PiPi stacking interaction. And so usually so empirically, so people know that aromatic rings, people observe that aromatic rings tend to bind together, stack together closely. And experimentally, so the stacking interactions are measured to be in a range of 2 to 3 kilocalorie per mole. So which is quite high. And so it is still under debate. So it is not completely understood why these two aromatic rings are interacting with each other, but because it's a complex phenomena actually. So coming from some dispersion interactions and also some quantum effects. But phenomenologically, so the two aromatic rings, it is clear that two aromatic rings are interacting with each other, attracting each other. So this PiPi stacking interaction is one of the most important forces determining the structures of biomolecules such as DNA or RNA. So we've talked about the scoring functions, and let's talk about how to sample, how to find, how to consider multiple ligand conformations during docking simulations. So there are many different algorithms to consider ligand flexibility. So the one of the example is called the anchor and grow algorithm. And this anchor and grow algorithm is used in the UCSF doc program. So we're going to practice this program, during the class. So in the UCSF doc simulation, one ligand is divided into substructures, and so these rotatable bonds are cut. And it is, cut into several pieces, several fragments. So which means that a ligand is broken by its rotatable bonds, and once they are broken, and the largest fragment is considered as an anchor. Okay. So when there is a protein, largest fragment is placed first, and then once anchor is bound stably, then the next fragment is bound here, and then the energy of each fragment is calculated. Then if you, in this example, and this fragment has a minus 20 kilocouple, and this fragment has minus 12 kilocouple, but this fragment has minus 3 kilocouple. So this one is discarded, this one is removed, and then so we have two possible structures. So this one is removed, and from here so we add the last fragment, then the whole conformation is reproduced. Then after that so we calculate energy of this, and we calculate energy of this, and we assume that lower energy so if E1 is lower than E2, then we believe that this conformation, this bound pose is more probable. So the ligand must be bound in this structure, in this pose. So that's our how we generally think about how we predict protein ligand bound structure. Okay, so this is an example of anchor and grow algorithm. So the name anchor and grow algorithm comes from the largest fragment is considered as an anchor, so it binds first, and then the whole ligand structure is grown to form the complete ligand structure. So this is one of the example of sampling multiple conformations, and actually so there are many other programs, and each program uses their own different approaches to consider the flexibility of a ligand. So so during the class so it's not possible to talk about all these algorithms, but during the class in the next class so we're going to practice actual protein ligand docking simulation, and by running the actual protein ligand docking simulation, and you'll see you'll get a hint, you'll know how the whole procedure works, and maybe you'll be able to run your own protein ligand docking simulations. Okay, so let's call it a day.